Good morning, everyone, and welcome. As she said, this webinar is being recorded. If you have any questions, you can put them in the Q&A and we'll try to get them answered. Thank you for joining us today for a discussion on impurity identification. This is a topic that can look very straightforward from the outside. You see an unknown peak, you collect some data, and you identify it. But in practice, impurity ID can be one of the most challenging parts of analytical development. It requires strong chromatography, good mass spec and spectral interpretation, a practical isolation strategy, and often a fair amount of patience and scientific judgment. Today, I'm very excited to be joined by Dr. Yvonne DePorre. Yvonne is an analytical R&D scientist with AJNA. Yvonne and I have had a chance to work together through AJNA, and I'm really looking forward to letting everyone hear directly from her today. Before we get into the technical discussion, I'm going to ask Yvonne to tell us a little bit about her background, her role at AJNA, and how she became involved in analytical development and impurity identification. I'll kick it over to you, Yvonne. Thank you so much.
Thanks so much, Wendy, and thanks for inviting me. I'm really excited to present today. I'm an analytical R&D chemist for AJNA Biosciences. And AJNA is a botanical drug development company, which is a very specific kind of niche industry, which I'll get into later. But my background is in chemistry. So I have a PhD in organic chemistry from University of Michigan, Ann Arbor. I was focusing mostly on mechanisms and synthesis. I have natural product target. And developing catalysts towards the synthesis of natural products and complex structures. And after my U of M PhD experience, I started working in the cannabis industry in Michigan for the state-regulated testing. And we were kind of one of the first labs in the state to bring on pesticide testing and heavy metals and things like that. Before AJNA, I worked for Charlotte's Web for about 3 years, and Charlotte's Web is a hemp CBD company, and there's now a collaboration between AJNA and Charlotte's Web called DeFloria, and my role at Charlotte's Web has led into AJNA, and since we are a botanical drug development company, we have really a lot of analytical chemistry because we're not just isolating or synthesizing a specific natural product structure as a drug. We're actually extracting the full spectrum of a plant, such as cannabis or mushrooms, fungi such as mushrooms. And the whole point of it is to take the whole, kind of like the whole therapeutic value of the substance, because our philosophy is that these natural substances that nature has made, people have been using them for thousands of years, but they just haven't necessarily been able to be FDA approved. And some of that is because actually of their chemical complexity, because the FDA requires everything that is an approved drug to be fully understood and fully known and fully standardized, everything about the manufacturing process and where it's from. And with a plant, it's just much harder to do that because plants can be variable. They have genetics that respond to different environments. And it's really a big project, but it is achievable. And that kind of relates to the topic of our webinar today, because the concept of unknown peaks, it could be lots of different things. It could be maybe something that is formed in the processing. Maybe there's like a side product, maybe something is getting heated or converted into another natural product. Another thing that could happen is degradation products. You could maybe have one of your APIs convert into something else during shelf life testing, stability testing. So those are kind of things that you really need to be cautious of. But in botanical drug development, there's also something called a related substance which isn't necessarily a process impurity or a degradant. It's just a related molecule that is being extracted with your main molecules of interest. So, yeah, the thing about, lots of peaks is that's actually kind of just inherent to natural products and food itself. Like, for example, we recently analyzed a tea sample just as kind of a preliminary testing of an instrument, and we found 1,000 peaks just in this tea sample. So the reason why these peaks need to be the reason why we need to identify peaks is because we're making a drug. And, if you have different natural products that are in food and people have consumed these foods for thousands and thousands of years, they don't necessarily need to be identified because we have historical data that it's safe enough, and people also aren't relying on these substances to treat a disease. It's just a regular food product. So
yeah, what you're doing at Ajna then is going in and looking at all the different components. It's not necessarily impurity identification, but as you said, it's related substance identification or really component identification of your product, of your botanical product.
Yes, exactly. So it can be a little bit daunting at first before knowing what any of the peaks are. And you can't just claim that they're all natural or okay. You have to prove that they actually are coming from your starting material. But yeah, it's, if you have a specific API that you're extracting, you can kind of think about like all the other molecules that are required to make that API within the organism, and then also maybe some side products that could be occurring at trace levels with different types of enzymes. So like just kind of thinking about natural products and how plants and fungi make them. It's usually some type of sequential synthesis that enzymes are doing. So understanding the biochemistry pathway of the different enzymes that make your compounds of interest can kind of give you some clues and ideas to what else might be there.
So kind of as a first screen, when you've got an unknown that you're trying to identify, then for botanical drugs in particular, you're looking at what are the pathways for that the organism produces the constituents in there, right? And then what else could be in there? And then as you said earlier, I think it could be degradants. For small molecules, it could be the starting material. It could be degradant of the starting material. It could be a lot of different things.
Yeah. If it was a synthesis, it could be the starting materials of that synthesis, making sure that they're fully removed from the final product. So it is a little bit different between botanical drug and more traditional pharmaceutical. But yeah, this is kind of the whole reason why we're doing it, that we want to have those extra peaks, because
and we need to know what they are.
And we're putting the whole substance through clinical trials as a whole.
Right. Yeah. So what is the biggest challenge that you found in the work that you've done?
Yeah. I think
the characterization.
The biggest challenge is finding something that just doesn't have a reference material available for it. So like some of them are easier to identify if they have a commercially available reference material from a reputable vendor that's going to come with a C of A, purity, maybe some NMR data, some mass spec data to go along with it. But yeah, if that's not available for the molecule that you think it is, then it becomes a much more intensive and long process that could involve many different techniques. You could, we often isolate the molecules if we can't, if we're not able to figure them out by screening reference materials, we start to isolate them. And then, we're isolating them because we want to take an NMR spectra on them, because the NMR is going to give us a roadmap of the structure of the molecule. And it can be really tricky because there's a lot of structural isomers in natural products. Sometimes, like if you think about terpenes as an example, there's different classes of terpenes, such as monoterpenes, sesquiterpenes, and these all have a specific number of carbons and hydrogens, and it can be pretty difficult to distinguish between them by mass spec alone. It's definitely not impossible, but having the NMR in combination with the mass spec can give you more information about exactly what is connected to what in the molecule in its 3D orientation. The reason why, oh, sorry, go ahead.
No, it's okay. It's okay.
Yeah, the reason why it needs to be an exact match is because when we find an unknown, it actually can't be quantified until we have a reference material. It can only really be estimated based on other compounds in the mixture. And it can't actually be quantitated. And if your reference material actually isn't a match, then you won't really be able to quantitate it because it might not really match the retention time.
Right.
And certain isomers are okay. Like if it's an enantiomer, it doesn't really matter because usually you're not using a chiral column, so you don't really always have to have, a perfect enantiomer, which is helpful, but it definitely matters for diastereomers and structural isomers. So
Right.
Yeah.
So maybe we can kind of go through the isolation and the concentration of the impurities, the different processes you use for that, and kind of what the challenges that you see there.
Yeah. So it is quite challenging because they're not the major component of the substance. So it's quite difficult to target something that's not the major component of a mixture.
Because of the concentration?
Yeah. The concentration is quite low of these. It's kind of more on the range of like less than 1% in the substance. So it is there are certain techniques that you probably could isolate it in just one round of chromatography, but typically we kind of have to isolate the bulk substance from the unknowns and then kind of focus on a specific unknown. So usually it's like a larger scale, kind of like a bulk separation, and then at the end just really focusing on a specific compound. And usually it's a pretty small amount of material in the end that's collected. It's kind of
on the order of like a few milligrams.
Yeah, just a few milligrams. So it can be a little bit labor intensive, but it is necessary for some of our compounds. Yeah.
And so what is the process that you go through to do the separation?
It's a flash chromatography with different mobile phases. So separating the compound of interest from other compounds with a gradient of mobile phase. So kind of going from one end of polarity to the other end, kind of separating things by their relative charges from polar to non-polar, and also the reverse of that.
And then collecting those fractions as you go.
So the fraction collection, like we have different strategies for isolating the compounds, and it is a bit labor intensive, but if we're looking for like multiple at the same time, we can kind of work together on them and not just one at a time.
Kind of group it together like that.
Yeah.
I had an impurity that we were trying to identify, and we were collecting fractions from the HPLC. So we're doing the largest injection that we could, and then collecting those fractions. And, because we didn't have like prep LC or flash chromatography or anything like that, we weren't able to get a significant amount of it, but it was enough for us to be able to perform the identification. I remember spending, I think I spent about a week sitting in front of the LC collecting it as it came off. So
nice.
A lot easier with a fraction collector. I was the fraction collector there. So
yeah.
So once you're there and you've got it isolated, then where do you go from there?
Yeah, so once we deem it's of acceptable purity, we run NMR on the sample. So starting off with proton NMR, carbon NMR, and then usually a few different 2D experiments to trace the different atoms of the structure. So, and yeah, an NMR is it's like a giant magnet, and we don't have an NMR ourselves in-house at AJNA, but we outsource and collaborate with other labs for NMR as well as GC. So most of the work that we do at AJNA is LC mass spec and also UV detection, and then using other methods such as GC and NMR just adds extra layers of confidence to our work.
Okay, excellent. Can you give me some examples of kind of how you've gone through? Because I know that we work kind of iteratively with that process as we're doing the testing here, and you're providing samples to us. Can you kind of tell us a little bit about how that goes?
Yeah. So once we have confidently identified a compound and we have a reference material available, we will add it to the quantitative method, and then we can quantitate it in future batches. So also the obtainment of the reference material itself, that can sometimes require an investment in some synthesis. There may need to be some troubleshooting for the synthesis if the reported route isn't necessarily high yielding. You might need to optimize it. So yeah, this is kind of where the synthesis part comes into play. So a lot of analytical work to figure out what it is, and then once we're pretty confident, making it and then testing it to 100% confirm that that's the structure.
So the process kind of is isolating. You isolate it, purify it, check it maybe against the methodology that you have, make sure that you isolated the right compound, you have the same spectra and retention time that you had before, and then send it out really for structural elucidation and to try to figure out what it is. How often do you identify something and find out that there already is a reference material, or do you have to go get that made sometimes?
Yeah, well, it's kind of hard to say like exactly how often, because it's for a specific project. But yeah, like the project with DeFloria, it was approximately like maybe half of the peaks that were able to be identified with a reference material. And also but yeah, they're getting like more reference materials made is definitely a priority. So kind of reaching out to the
CRM manufacturers. We use Cayman a lot here, reaching out to them and seeing if they can manufacture that for you, and that's something that you can.
It kind of depends on if other groups are interested in the molecule as well.
Mm-hmm.
So if it's something that requires like multiple steps to make and it's going to be a really expensive synthesis, and if you're the only group that's interested in it, it's going to be pretty expensive. But if it's something that's maybe just one or two steps and there just happens to be other groups asking them for it at the same time because maybe it just was published recently, then actually it's not really too expensive. So it really depends on the difficulty of synthesizing the molecule, number of steps, the yield of the steps, and whether they feel like they would need to optimize those yields. Because sometimes something is just, it's been synthesized one time in the literature 30 years ago and it was a 15% yield or something.
Mm-hmm.
So sometimes you don't necessarily want to rely on that as a commercial process if something is like so low yielding.
Right. Yeah. So I would imagine that if it was like a new chemical entity, I guess there aren't a lot of people that are looking for that, so it would get pretty costly if you have a number of impurities that you need to get synthesized.
Yeah. Yep.
Yeah. So can you tell me if there's any kind of challenges that you've had in impurity isolation and identification?
Yeah. It's pretty challenging when you have isomers and some of the isomers are so close to each other that they actually seem like they're the identical compound if the method doesn't have enough separation. So for really truly identifying something, you want to have a lot of chromatographic separation, so you can see maybe some really subtle differences in retention time. So
maybe an orthogonal method, that has different specificity to really tease out whether that's the same one. I know we've had situations where, as we were working on some stuff where you had a retention time match in your lab, and then when we ran it on our method, we didn't have a retention time match. Like there's very slight differences that when we find out that maybe that we don't have that peak figured out quite yet. Do you have any advice?
It's also kind of interesting because a lot of compounds, they're usually reported as like either a natural product or something that's synthesized, and people kind of have this idea of like, oh, this is a synthetic cannabinoid or this is a naturally occurring cannabinoid. But you don't necessarily want to limit yourself to those categories when you're trying to figure out unknowns, because there just could be a lot of things happening in the plant that you're not aware of. So I would, don't just look at natural product papers, like look at synthetic cannabinoids and all kinds of literature and research, because you don't want to like limit yourself to only things that have been isolated from a specific plant. So like review articles are really helpful for like just kind of getting started, just looking at all the different compounds that have been isolated from cannabis, like hundreds and thousands. But it is good to keep in mind though that you're definitely not going to find all of these compounds. Like there's going to be a lot of negative screening results. So you kind of have to be ready for screening, and maybe you kind of have to like accept that not all of the compounds you screen will always be a perfect hit, and then you can still gain information from things that are close and make the next step in getting closer to identifying a compound.
Be prepared for disappointment, I think.
Yeah.
Yeah. Yeah. Be prepared for disappointment. Has there anything that you've done that's been like particularly interesting or rewarding in the identification work that you've done?
Yeah. I think like it was almost 2 years ago that I joined AJNA, and yeah, we were about to submit our botanical drug application to the FDA, and we just really had to figure out 2 peaks to be able to move forward. And yeah, the first one we got by screening, and the second one was the difficult one that we were talking about before, that matched with us but then didn't match at Triverity. So it was like a pretty it was like we were kind of, this was kind of like one of the last things that we had to complete to submit everything. So I think like figuring that out so that we could move forward was really rewarding. So
yeah. It's always good to solve a problem and to get the answer that you're looking for. Is there something that, when you're doing impurity identification that takes longer than people would think? I think, as I said in the beginning, we're approached a lot for impurity identification, and people think that we can just, run it on our HPLC or even run it on an LCMS and be able to figure out what it is right away, right? That it's just essentially one injection and we'll know what it is. And that's really not the case. Like, what is it that you think like takes longer than people would expect?
Probably like the interpretation of the data.
Okay.
Just like kind of really finding the time to sit down and just really, really look at the fragments of the mass spec data, drawing the structure, and thinking about what functional groups could be responsible for the peaks that you're seeing in the targeted mass spec. So yeah, there are a lot of tools that you can use to use your mass spec data and match it with compounds. And that actually can be, like you actually can sometimes figure out an unknown peak in a day if you can match it to a spectral compound and it makes complete sense within what your sample is. But if it's not that, then first of all, the whole process of isolating it is pretty long. That's usually at least a month or two, sometimes longer if you're working on other things. So usually 3 months or more. But yeah, like having the data and just really analyzing the data and taking the time to think of ideas that could be happening. So, because it can just be overwhelming when there's so many different compounds and you're like, oh, I should isolate that one. And like, there's so many different things to do. Sometimes it's hard to just look at maybe just 1 or 2 pieces of data for like a whole day or something. So,
right. And I think then integrating the mass spec and then the NMR data together to understand the structure of the molecule. And I think that's really interesting that it would take 2 to 3 months for isolation of an impurity, right? 2 to 3 months to get yourself a few milligrams. Those milligrams would be pretty precious then, I would guess.
Yeah, like the material is like, once you kind of get to the end of the purification, I'm usually being really cautious with the material, making sure I don't spill it or drop it in the rotovap or something like that. That's kind of like the last thing you want.
Devastating.
Ruin your sample right before it's almost ready. So yeah, just kind of being really careful at the end, just taking it slow, not rushing. But yeah, it's kind of one of those things where once you kind of get your compound as the major component, you can kind of see the end and know that it's going to get better, and that you probably will maybe only need 1 or 2 more purifications to get it to the final purity. So yeah, at the beginning, it can be hard because it just kind of seems like you're looking at, so many peaks, and it just seems so messy. But once you kind of get it as, either your major peak or one of your major peaks, it's a little bit more, you can kind of see that it will be purified and it's possible. So yeah.
So I would imagine,
and yeah, it's also kind of hard it depends where they are in the chromatography. If they're right next to the major cannabinoids, it's going to be more difficult than if they're at a different part of the spectrum, if they're like maybe at the very beginning or at the very end. So
I would imagine then if you've got an impurity or a component that's at a fraction of a percent, trying to get a few milligrams of that, you have to put a lot of material then into that system and purify a lot of material to get to a few milligrams.
Yeah, we usually we have quite a lot of material to use usually, because we're making like all these batches for the purpose of characterization. So it's usually not necessarily an issue of not having enough material. It's just like being able to scale up the process. So yeah, we just don't have like the, really, really large scale purification system.
So you're doing a lot of
like large scale purification yet. So
yeah, so a lot of injections to get the material through maybe a first pass, and then you're collecting that to then refine it down, right? Yeah. There's SFC, which uses supercritical CO2 as the mobile phase, kind of replacing kind of trying to replace heptane in terms of its polarity. And we've used that a little bit. But yeah, it can be a little bit challenging. There's kind of, different techniques that work for different types of compounds better than others. Yeah. So is there anything that, as methods are being developed, because we're analytical development here, as methods are being developed, is there anything that you can do to kind of help with impurity characterization?
Yeah, well, actually the method that we use UV in tandem with mass spec. So I think it's really helpful to have both together, because
you can get 2 data points then, really.
Yeah.
Well, really 3 with the retention
spectrum
time. Also,
the UV spectrum can kind of give you a hint of the functional groups in the molecule. So if it looks pretty similar to other UV spectrum, it might be that group of molecules, like the cannabinoid acids and the neutral cannabinoids kind of have a different types of peaks in the spectrum. So yeah.
So it's another piece of information, another piece of data to help with identification. We had a few questions come in on the chat. The first one is, what type of NMR do you use to determine the structure?
Thanks. Yeah, we collaborate with a lab that has a 400 megahertz with a cryoprobe. So the cryoprobe has been really, really helpful to increase our signal in the proton NMR. So since we have a limited amount of material, the more sensitive NMR techniques are much better for us. So
what is the cryo? Can you tell us a little bit about? I'm not familiar with a cryoprobe.
Yeah, it cools down it cools down everything to about 20 Kelvin with helium gas. So and then when everything cools down, the signal to noise ratio is much higher. Yeah. Because the NMR is basically this giant magnet. It's like an MRI machine for molecules. So you're like putting your sample inside the giant magnet. And cooling it down makes the signal to noise much higher. So there's a lot of quantum physics behind NMR, with the spin multiplicity of the different atoms. So that's why carbon-13 is less sensitive than proton, because they have to use carbon-13 instead of carbon-12. So it's less abundant. Therefore, usually you need to run carbon for a longer period of time, maybe like overnight or for a day,
to be able to get
that nuclei is less abundant. So
yeah. The next question was, aside from NMR and mass spec, what are the other techniques that can be used to elucidate the structure of unknowns?
There's also, a few like qualitative tests like stains. We don't do that very often, but like kind of like staining a TLC plate with different solutions can help give information about the functional groups.
Do you use TLC?
And then also the UV spectrum.
The UV spectrum
with the MS.
Yeah. Do you ever do any like XRD or anything like that?
No. Well, we don't have enough material to crystallize. But we've definitely thought about it. And maybe once
we haven't needed to,
we get one of ours, maybe one of them that's like pretty novel, we would get a X-ray structure once we have this reference material made. So
and the other one we have is, after you have identified the reference material or an impurity, do you have to study its degradants and impurities as well, according to the FDA regulations?
Yeah. So there's like a whole FDA guidelines called the Botanical Drug Development Guidelines. And most of my work is relating to chemistry, manufacturing, and controls. It's called CMC. The FDA regulations state, the guidelines state all of the requirements, but it's also kind of like a working relationship between your group and the FDA, because every time you submit something, they're going to have questions that you need to address. And it doesn't necessarily matter if those things weren't in the original guidelines. The guidelines are just a framework and not an official final guideline. And it says that in the document that everything is non-binding.
Well, and I think with the botanical drug guidance being so new, relatively so new, right? There's a few submissions now, and we're seeing a little bit on how the FDA is approaching botanical drug characterization and all of that kind of stuff. And so it's, I think it's going to be really interesting over the next few years as those drugs progress through the approval process to see, and ajnas as well, and see kind of how the FDA, it's exciting to have this pathway for botanical drugs. I think the question is, when we've identified the impurity, do we have to look at the impurities? I think it's do we need to look at the impurities, or are we really just looking at that impurity? And I think the answer from my point of view is we're just looking at that impurity. It could be that impurity has some impurities that then we would characterize separately if they are at a level that we can detect them and they're above the threshold above which we need to identify them. But I don't know if you have anything else on that too. I think we would treat it as a separate impurity. and not really
Yeah,
consider.
yeah. I think like it really just depends on what you find. Like if the substance is found has like a clear connection to your the activity of your API, if you think if you have scientific evidence that it's directly contributing to the efficaciousness of your API, versus maybe just a related substance that it's not necessarily good or bad. There's not necessarily evidence that it's working with it's and not necessarily the most active component. But yeah, there's different categories. So something that was originally found as an impurity could actually become something that is quantitated and tracked in the batch. So it just kind of depends how important it is. If it's important enough, it'll be there will be a spec set for it for how much there should be in each batch. But if it's not really that important, then it's just named and just.
I think, yeah, and I think that it depends a little bit on its toxicity, if it's particularly genotoxic impurity, or like, as you said, it's kind of a component, not necessarily an impurity, then it needs to be measured more. To kind of like circle back around, we've talked a lot about NMR and mass spec and UV and all of these different things. When you look at your graduate work and your background, what really gave you the tools to be able to do the work that you're doing now?
Yeah, I guess not necessarily like any one lab or experience, but just having a variety of different lab experiences. I went to Michigan State for undergraduate, and I started doing research in biochemistry with Dr. Walker, some enzyme chemistry. So yeah, my first research experience was learning about enzyme assays and helping with enzyme purification. So yeah, I would say like probably my very first undergrad lab was really helpful to just start thinking about enzymes and biochemistry, even though it's not necessarily my major. I also did some inorganic chemistry, and yeah, that's really good for thinking about mechanisms, reactivity, and yeah, also I've done some computational chemistry before, so that's pretty helpful for thinking about how we could maybe optimize things and try to use our data to obtain the structure. So
yeah. And I think when you look at impurity identification, it really is, there's a lot that goes into it. It's not unfortunately, sometimes, as you said, it's very straightforward in that you can get a hit right away. Maybe you get a hit from a library. It's a really well-known compound, and it's in a mass spec library, or you've got experience enough to know that this is a known impurity or a known degradant, or there's some literature data, and it's pretty easy to figure out what it is. But sometimes you just really have no idea, and doing the work to isolate it and purify it and then send it out to get, or maybe you've got an NMR on site, to get the NMR done to be able to determine the spectrum, really is a long and costly process if we don't have somebody like you on staff, right? So it takes a long time. Is there anything else that we haven't covered today that you wanted to cover?
I don't think so. I think that's about it.
Okay, awesome. I've got a thing here. So thank you so much for joining us today, Yvonne. I really appreciate it. Thanks for sharing your perspective. I think this discussion really highlights that impurity identification is not just checking a box. It's scientific investigation that requires the right tools, experience, and strategy. For those listening, I hope this gives you a better appreciation for why unknown impurities should be addressed early in development before they become stability, specification, or regulatory issues later on. At Triverity, this is exactly the kind of analytical development challenges we help clients work through, whether that means evaluating unknown peaks, developing better methods, isolating impurities, or supporting the next steps towards structural identification. Thank you again, Yvonne. I really appreciate it. And thank you everybody that that joined us today. We appreciate your time and look forward to continuing the conversation.
Yeah, thank you so much.